So hi everyone, my name is Wilson Fandino. I'm a candidate for senior TA for this year, but I've been collaborating with the PPCR course over the last 4 years. Basically, to talk to you a little bit about myself. I'm a consultant anesthetist working here in London, and I'm strongly interested in the statistical part of the course, so I will be hopefully involved in quite a few lectures, especially the ones related to biostatistics. So in regard to the assignment for this TA PPCR workshop, I have decided to deliver a short presentation on the common distributions that we normally use in biostatistics, and have decided to talk about this topic because it's a little bit, I would say it's a bit complex, but considering that this is same for senior TAs, so it might be interesting and helpful for most of you. So I hope you enjoy my presentation. And so basically, I'm going to talk about, before talking about distributions, I would like to talk about 4 important functions in biostatistics, which are the probability mass function, the probability density function, the cumulative distribution function, and the survival function. So I will talk very quickly about all the, as I say, the most common distributions, including the binomial, the normal log normal distribution, the chi-square distribution, hypergeometric distribution, which is important to discuss the Fisher test, the sampling distribution, the students' T distribution, the F distribution in the context of ANOVA, the parametric Weibull distribution in the context of survival analysis, and finally the beta distribution, which is important to understand the Bayesian analysis. So basically, this is the classification that we use in PPCR, and I think it's a quite neat classification of the variables that we use in clinical research. So basically, we like to split these variables in numerical, categorical, and ordinal. So as you know, the categorical can be dichotomic or non-dichotomic, and the numerical can be continuous or discrete, depending on whether the variable can take decimals or not. But from the statistical point of view, the random variables are basically divided into only 2 groups. So the random variables from the statistical perspective can be random continuous variable or random discrete variable. So you know how to differentiate the interval and ratio of variables when we are talking about the continuous variable. But I just brought this slide to emphasize that purely from the statistical perspective, there are only 2 random variables, which are the continuous and the discrete ones. So based on these concepts, so whenever you have discrete variable, so the function associated with this random discrete variable is precisely the probability mass function, which is a very important concept in biostatistics. And on the other hand, the function associated with a random continuous variable is the probability density function. So as you can see here, the distribution is quite different. Well, it might remind you the shape of the bell curve, but it's not exactly, because you see that the variables are discrete, so they are not taking any decimals. They can only take integer values, sorry, and the function associated with this probability mass function is exactly these ones. As you can see here, I'm not going to explain to you in detail this because it will take me a long time for this presentation, but I just wanted to make a difference between the probability mass function and the probability density function. As I said before, the function associated with the PDF is quite different because it's associated with the random continuous variable, and you see here the z-score, which we are going to discuss later on in this presentation. And then so basically the PDF is just the area under the curve, the probability of the area under the curve when the z variable takes any value on the horizontal axis here. So let's say, for example, if the z takes a value of 1.96, which is here, so the area under the curve here is going to be exactly 2.5%, and we are talking about two-tail test, so obviously we have to take into account the other 2.5%, which is exactly here in the minus 1.96 point of z. So apart from the PDF, there is another function which is a cumulative distribution function, and basically the cumulative distribution function is this is what I'm representing here. And by the way, all these graphs have been designed based on the Stata software, so you can actually reproduce this one as all of these graphs that I brought to you for this presentation are made with this data. So I'm not using any other funny software. And I was mentioning that the CDF is a little bit different from the PDF, because the PDF is the area under the curve, and then the cumulative distribution function is the probability that the x value, which is here, would take more than that value, that the x value. Say, for example, in this case, so the probability of having more than 0 value is going to be 50%, as you can see here. So this is what the CDF tell us whenever you see this. And then there is a relationship between the CDF and the survival function. So basically, as I said, the CDF is the probability that the random variable take a value of more than 1, or more or equal than 1. So in this particular example, that possibility is going to be like 84 or 83%. And then the survival function is exactly the contrary of the cumulative distribution function. So it's defined by 1 minus CDF. So in this case, if the CDF is 0.83, so the survival function is going to be exactly 17%. So the CDF is the possibility of having more than a value more than 1 in this case. And then there is another important distribution, which is the binomial distribution, and it's exactly what happens when we flip a coin. So this experiment is actually quite important in statistics, because many of the experiments in clinical research, obviously we are not going to flip a coin, but they are based on this concept, on the concept of flipping a coin. For example, the fact that if we want to categorize a variable or dichotomize a variable like hypertension, yes or not, so the fact that the patient has or not hypertension, say no hypertension is 0 and having the high blood pressure is 1, so this problem is related from the statistical point of view of with the problem of flipping a coin. So that means that we flip a coin and it's head, so the head would be hypertension, and if it's tail, so the tail will be no hypertension. So this is what we normally how we normally interpret this from the statistical point of view. So this is why it's important to discuss this, and this is again so this is the binomial coefficient, and this is the actual binomial distribution I use as I explained to you here. So there is a binomial coefficient, which is this represented by this notation, n choose x, and the n choose x is exactly this one, where n is the number of trials, x is the number of successes, and this notation here is the factorial. So this is the binomial coefficient, and if we multiply the binomial coefficient by the Bernoulli distribution, which is exactly this one, we will obtain exactly the probability mass function for a random discrete variable. So the Bernoulli distribution, by the way, is represented by this notation, p to the x multiplied by 1 minus p to the n minus x. So where the p is the probability, in this case the probability of having a head or having a tail, and 1 minus p is exactly the contrary. For example, if the probability of having a tail is 0.5, obviously the 1 minus p is going to be 0.5 as well, but let's assume that you are having a biased coin. So if the probability of having a head, for example, say is 0.3, so 1 minus p is going to be 0.7, And again, the x is the number of trials, and n is the number of the n is the number of trials, and the x is the number of successes. So this is how we normally represent the binomial distribution, which is exactly this one. So this is what I was trying to explain to you a couple of minutes before: the number of attempts, the number of hats, and the probability of success. So in this case, if the probability of success is 0.5, in other words, the coin is fair, and the number of attempts is 10, okay, so the possibility of having 1, 2, 3, 4, 5, 6, 7, 8, 9 or 10 tails, 10 heads, is going to be this, the probability. So as you can see here, the probability of having 5 heads out of 10 attempts, surprisingly, it's rather low, because I mean, intuitively we could say, it could be more than 50%. So based on this theorem, the probability of having 5 heads and 5 tails is going to be only 24%. So as I say, this is a very important concept in biostatistics, and this is called the binomial distribution. And the binomial distribution, by the way, it might remind us the shape of a bell curve, but it's not exactly the shape of a bell curve, because the kurtosis is less than normal distribution, and the skewness is obviously very symmetric, but the shape of the curve is much taller and much tight than the normal distribution. So this is the normal distribution. So what you can see here is just the PDF for in order to construct this curve. So this is the function of the curve, and then so you can see here the description or the notation of the normal distribution in terms of statistics. So basically is E to the z square over 2, and this is all divided by the square root of 2 pi. So many of you may wonder where these numbers came from, because it's actually quite interesting, because why or how do we calculate that? Obviously this is the 50%, but when we are talking about one standard deviation away, so we are talking about the 68% when it's two-tailed, and when it's 1.96 standard deviations away from the mean, so this is going to be exactly 95%. But how do we realize, or how do we calculate this? How do we estimate this? So basically this is the area under the curve, and this is why it's important to understand the meaning of the PDF and the CDF. So in order to calculate the area under the curve of this bell curve, we have to use integrals, because the area, obviously, I mean, you can calculate, for example, the area of this rectangle, but because this is a curve, it's quite difficult to calculate exactly the area under the curve of this because of this curve. So this is why we have to use the integral, which is exactly the CDF, and this is how we calculate the area under the curve with the CDF. So based on this, so many of you have seen the Z-score, which are actually very important as well in biostatistics. And the Z-scores are nothing else than the area under the curve, as I was explaining to you. So, for example, so this is the actual Z scores table, and I'm going to try to explain to you how to use this. I already said that the CDF is the area under the curve. So let's say, for example, that you are interested in the area under the curve for 1.96. So you just need to look at the 1.9. So the column here is 1.90. This is corresponding to 9.91, and then the 9.96 is exactly this value, which is 0.975. So this means that 0.975 is this area. This is the area under the curve. Okay. So because we are not interested in this area, but we are interested in this area, this and this, when we are talking about two-tailed test. So all we need to do is just because it's 97.5, so that means that this is going to be 50% for sure of the curve, 50%. So we can subtract this 50%, and then we can say that 47.5% of the curve is represented by this green area. And then if 47.5% is this area, so that means that 95, which is 47.5 times 2, is represented by this area. And this is why the 5% is represented by 2.5% here and 2.5% here. And this is the way we have to interpret the Z-score. Another example for one side test is exactly 1.64. So if you look at this 1.6, so 1.64 is here, which is exactly 0.949. Rounding is 0.95. So that means that 0.95 is here, and if 0.95 is here, so that means that the 5% under the curve is corresponding to this area. So this means this also means that that any value more than 1.96 or 1.64 in one side test is going to correspond to less than 5%. So in that case, we are going to reject the null hypothesis. And then there is another distribution which is called the log normal distribution. There's usually a confusion in regard to what exactly is the log normal distribution. So what you can see here is the log normal distribution represented in red. And so basically this is not a normal distribution, obviously, but if you take the logarithm of these values, you will obtain normal distribution, which is a transformation, a way to transform our data. There is another very important distribution in clinical research, which is the chi-square, obviously. And I say it's really important because many of the tests that we use are based on or are following this distribution. And I brought some examples here in this slide. So, for example, the McNemar test, which is basically the version for matched or for pair data of the chi-square. The Cochrane-Mantel and Hansel test, the CMH test, which is very similar to the McNemar test, but it rather than pair and test, they are this test can accept more than two variables, for example, ordinal variables. And then the Friedman test, which is the non-parametric version of the repeat measures ANOVA. And Laurent test, which is actually quite important in survival analysis to test the hypothesis of the survival, and then the likelihood ratio test, which is also very important when we are talking about logistic regression, to see how good our model is fit to our distribution. So as you can see, many of the tests that we use in clinical research follow a chi-square distribution. And you can see here the PDF function, which is a little bit complex, so I'm not going to go in detail for this, but I use this equation to represent the curve for the chi-square distribution. So again, this is done with Stata. And what you can see here is the value of the degrees of freedom. So when we got a 2x2 contingency table, so obviously we got 1 degree of freedom, so this is going to be the distribution. And you can remember from the chi-square test, 3.84 is the critical value for a 5% of alpha value. So this means that the 3.84 is exactly located here, and this also means that the area under the curve is represented by 95% of the area is here, all this, and this is going to go even higher, and 5% of the area is going to be represented here, but by this very small area. So this actually help us understanding the how to reject the null hypothesis, or how to fail to reject the null hypothesis, because if you got say a chi-square value of 4.7, so it's going to be here, so we already know that is within the 5% of the area, so we can reject the null hypothesis. But if you've got say 2.7 for always, obviously for 1 degree of freedom, for example 2.7 is here, so we know that is not into the 5% that we are interested, the area of the rejection of the null hypothesis, so we fail to reject the null hypothesis. And some more examples here for 2 degrees of freedom, for 3 degrees of freedom, and for 4 degrees of freedom. And you can see that for 2 degrees of freedom is a little bit similar to 1, but it's, as you see here, it's not exactly the same. But when you got 3 degrees of freedom, the distribution changes dramatically, same as with 4 degrees of freedom. So I think it's interesting and very useful to know these things. And this is something that I developed as well to try to explain the p-values actually. So I'm representing here the p-values, and what you can see here is the degrees of freedom. So again, the 3.84 is here, so this means that the 5% of this curve is represented by this. And then you can remember for from the chi-square test, the critical value for for 2 degrees of freedom is exactly 5.99. So this is represented by the red curve here. So you can allocate the 5.99 is going to be here. So that means that 5% of this curve is going to be represented here, and 95% of this curve is going to be represented by all of this. Okay, and so on. So you can do the same exercises with the 3, 4, or 5 degrees of freedom. There is another interesting distribution which is called hypergeometric distribution. So basically, this is actually why related to the binomial distribution, and I think a nice way to explain to you this is try to imagine an experiment where we got a number of, for example, black balls, we got another number of white balls in an urn. Okay, so so these are the possibility of drawing X number of black balls and another number of white balls. So basically there are two ways to think about this experiment. So when you are having the balls in the urn and you are replacing the balls whenever you are picking the black or the white, so you are replacing there back to the urn, or if you are not replacing those balls. So if you are replacing the balls, we are talking about binomial distribution. But if you are drawing the balls, whatever the black or white balls are, but you are not replacing those balls, you are not taking back the balls to the urn, so we are talking about the hypergeometric distribution. And it's quite interesting because again, I'm talking about the binomial coefficient here, and this is a way to explain the calculation of the feature exact test. Obviously, we don't need to do this in reality because Stata is doing everything for us. But again, I really think it's interesting to know how Stata is doing this. So how to calculate manually the feature exact test. And obviously it becomes more and more difficult when the sample size increases, but we can actually calculate this when the sample size is reasonably small. So again, I'm talking about the binomial coefficient here, and this might be new for some of you, but basically this is the notation to describe the binomial coefficient, and this should be read as because actually this is not denominator here. There is no numerator or denominator here because I'm not putting the it's not n over x. This is what I mean, but this should be read as n choose x. Okay, so n choose x is equal to n factorial over x factorial times n minus x factorial. And by factorial, I mean, for example, if you say there were 5 trials or 5 attempts, in this case would be n equal to 5. So 5 factorial is equal to 1 times 2 times 3 times 4 times 5, and so on. So this is the way to calculate the binomial coefficient. And then the probability mass function that I explained to you before is defined by this funny equation: x choose x, n minus x choose n minus x, but in this case is lowercase, over n choose n. So if you look at the notation here, it's quite easy to calculate the factorials, because all I'm doing here is just describe this notation in my equation here, and this is the probability mass function. So by doing this, obviously you replace this. I'm not going to talk about x anymore. I'm going to talk about a, b, c, and d, and then there's going to be exactly this: a plus b choose a times c plus d choose c over n choose a plus c. So if you do this, it turns out that the equation for the calculation of the Fisher exact test is exactly this one, which is a plus b factorial. So this number, whatever it is in your 2 by 2 contingency table, plus this number factorial, and then times c plus d factorial, and then times a plus c factorial, and then times b plus d factorial, over n factorial, which is here, times a factorial times b factorial times c factorial times d factorial. So this is what Stata actually does for us, and this is why it's taking quite a long time when you are plugging 2 by 2 contingency table with a very large sample size. So imagine, for example, that here there are 700 patients in A, in the cell A. So that's going to be, for example, that in B there are 300 patients, so that's going to be 1,000. So A plus B factorial is going to be 1,000 factorial. So that means 1 times 2 times 3 times 4 times 900 times 999 factorial. So this is quite difficult to calculate in Stata, even for the computer, and this is why sometimes Stata can actually be stuck when you are plugging numbers as large as them, and this is why for small sample sizes you will see in statistical books that for sample sizes less than 200, for n less than 200, they use the exact Fisher test, and for more than 200 they use the chi-square test. This is explanation, but we learned from our PPCR course that we should use the Fisher exact test whenever we can. If Stata is able to do the Fisher exact test, we should report this in our paper, because this is the exact calculation. The p-value reported from the chi-square distribution is just an approximation, and the p-value of the Fisher exact test is as the number suggests, is exactly the p-value for that distribution, for that hypergeometric distribution. So this is the probability mass function that I designed with the state program as well, just to try to make some difference between the number of successes when the number of trials is 100 in this example, and the number of heads or tails is going to be 50 out of the 100. So the number of successes, the probability of the number of successes is quite variable depending of what we are talking about. So when the number of successes is 10, is the distribution is actually quite different from n equal to 20 or n equal to 30. So again, the distributions are very different depending of the number of n, and this is basically the reason I brought this slide for you. So this is actually the calculation of the Fisher exact test based on the hypergeometric distribution. So this is what I explained to you a couple of slides before, and I'm using here obviously very small numbers in order to facilitate the calculation, and so you can see here, for example, that the p theoretical is in this case 8, which is here, 10 factorial, which is here, 9 factorial and 9 factorial, and you can see here the denominator as well. So the p-value is going to be exactly this p-value. And then the calculation comes from these theoretical tables. So you can see here the experimental tables, but you can also construct a theoretical table based on the possible values that the A cell can take. So if A plus B is 8 is equal to 8, which is here, so that means that the values that A cell can tell are only these 9 values: 0, 1, 2, 3, 4, 5, 6, 7, or 8. There's no more possibilities. So once you define this value, and provided that the marginals are fixed here, which are all fixed, so you see that the numbers are exactly the same here, 9, 9, and 18 for all the theoretical tables. So you can actually realize, or you can actually calculate what would be the values for the V, C, and D cells. And then so basically the calculation comes from the extreme values, what would be the more string table that we can obtain. And in this case, the more string tables compared to the experimental table are exactly these ones: this one, and this one, when we are talking about the two-side test, because you can do a one-side test, Fisher square as well. So what I'm doing here, here I calculate the P experimental, which is based on these values, but here I have calculated the theoretical values as well, which are based on these theoretical tables, on this one exactly. So you can see here 8, 10, 9, and 9. So it comes to it works out to be 0.00021. So so for the one side, p-value for the feature exact test is going to be exactly the sum of the p of the experimental plus the p of the theoretical, which is obtained from this table. And if you want to do a two-tailed test, so you just need to multiply this value plus this value times 2, which I did here, and this is going to be exactly the p-value. So in this case is 0.015, which means that obviously we have to reject the null hypothesis because it's less than 0.5. There is another distribution which is called the sampling distribution, and I brought this because it's interesting to understand a little bit more the concept of the central limit theorem. Basically, this is what we are doing in clinical research. So we've got a target population here, and then we got a sampling population. So we are just picking up some random patients from a target population, provided that all these patients have the same opportunity to take part of the clinical trial. And this is the sampling population. Let's imagine that you go here for 10, you got here 100 patients from the sampling population, and you are taking 4 then randomly in this example. So when you're doing this over and over and over, obviously you are not going to do this, but this is more a theoretical concept. You do this over and over, let's say 1,000 times. You take these 4 patients, and then you take another 4 patients randomly from the same target population, and then again 4 and 4 and 4, and you take say 1,000 of times. So you will obtain a normal distribution like this based on the mean values of these sampling of these samples, basically. So this is why the central limit theorem, when we got enough sample size, so we say that the population parameter for the mean, for the standard deviation, and for the size is the mu, the sigma, and the n capital letter. So this is what we are actually interested in, but we don't have these actual values. We do have the mean of our sample size, we got the standard deviation, and we got the sample size of our research study. So from the statistical point of view, so the sample statistics, the mean of the sample statistics is an estimation of the population mean, same as the standard deviation of the sample size is an estimation of the standard deviation of the population, and provided that those samples are taken randomly, number one, and provided that the sample size is large enough, in this case more than 30. So this is the sampling distribution. And then obviously there is another interesting distribution which I need to talk about, which is the student's t distribution. Again, this is very similar to the normal distribution, as it was the binomial compared to the normal distribution, but in this case the the function of the curve is actually quite different. It involves some complex distributions or complex function, including the gamma function, which is represented by this Greek letter, gamma function. And based on this function, so I have represented this in its data. For your in order for you to compare this distribution, I brought here the normal distribution, and so you can compare the shape of the distribution when we are talking about the T distribution, which are these two graphs here on the left, and this is the normal distribution. So you can see how different is the function. Actually, I did explain to you this function before, which is very important to, as I say, to represent the normal curve here in a graph. But it's also important to understand the z-score, as I explained to you. And then you can see here that it's quite different from the function of the T distribution, but in any case it's very similar, the shape of the curve. And it's quite interesting because what you can see here in this example is distribution with 3 degrees of freedom, which means that the sample size is only 5 patients. And then you go here distribution when the sample size is 30 patients. So you see here that the when we have only 5 patients, so the shape of the distribution, the shape of the curve, is actually quite different from the normal distribution. So that's why we can draw any conclusions based on normal distribution. But if you increase the sample size, in this case the sample size is 30, so it's actually quite similar. It's really similar to the normal distribution, despite the fact that the function is completely different. So this is why we say that when the sample size is large enough, so the student's distribution approximates to the normal distribution. So this is the another way to explain the central limit theorem. So provided that the variable is normally distributed, the more you increase the sample size, the more similar to the normal distribution is your T distribution. And this is why I brought for you here the T table. So for a two-tailed test, you won't find any more values more than 120. Whatever you read, I mean, this is easily available in statistical books or in internet. So all the T tables are provided only up to 120 degrees of freedom, and the reason is because any sample size more than 120 is going to follow very approximately to normal distribution. And you can see here that the T value for 120 degrees of freedom is 1.97, which is quite similar to, and then 1.96 from the normal distribution. And then if the, as I'm representing here, if the degrees of freedom tends to be to the infinite, so the T value is going to be exactly 1.96, which is exactly the one we use for the Z-score, the one that I explained to you before. So this is another representation of what I was trying to explain to you a couple of slides before. And then what I'm trying to say is that the shape of the curve is actually quite similar, but you see here for in this case for n equal to 4 is actually different. Is different from for example n equal to 30. And then there is yet another distribution, which is the F Snedeker distribution, and this is the distribution followed by ANOVA, as many of you remember. And this is just the probability density function that I use for again for the representation of the distribution in this data. So it's actually a little bit more complex, and as I say, involves some really complex functions, include the beta function and the gamma function. So the gamma function is here, and the beta function is represented here. So it's actually quite complex, but I managed to represent this in this data, and this is what I obtained from this PDF. So again, you see how different is the distribution depending on the degrees of freedom. So in this case, the K is the number of groups. So for example, you've got here 3 groups, and then the degrees of freedom are represented here. So when you have, for example, 3 groups and 20 degrees of freedom, you have to follow this this curve, and then if you increase the number of groups here in this case, so you see how different is going to be the curve. And again, I really think this is helpful to understand why certain value under k equal 3 is significant, and why some other value is not significant, and the assumption that there are more groups here in this case. As I say, the k is equal to the number of groups. So again, this is another example, another extreme example. In this case, what you can see here, the 3.48 is based on this table, F Snedeker table distribution, which is for 9 degrees of freedom and 5 degrees on the numerator, 5 on the numerator and 9 on the denominator. Okay, so you see that distribution is quite different from this one, and the distribution I'm using here is based on 2 degrees of freedom in the numerator and 57 degrees of freedom in the denominator. So that means that if there are 2 degrees of freedom in the numerator, that means that there are 3 groups, and if there are 57 degrees of freedom here, that means that the total sample size is 60, because the degrees of freedom in this case is n minus k. So that means that here we are talking about 3 groups consisting of 20 patients each, and you see how different is the distribution here. So this is what I was trying to say with this slide. And then there is another distribution which is called Weibull distribution. You may remember this from the survival analysis. So basically, this is you have to remember that the Cox model is a semi-parametric model, but there is an alternative version of this model, which is the parametric version, and this is the Parametric Weibull distribution. So again, you got here the PDF. And this is actually a little bit more complex because we had to take into account the three parameters, which are the shape parameter, the scale parameter, and the location parameter. Again, I'm not going to explain in detail because I don't think this is the scope of this presentation. But basically, when the location parameter is 0 and when the scale parameter is 1, so we are talking about standard Weibull distribution, which is represented by this function, and is nothing else than the hazard function, which is represented here, times the survival function here. So you've got here on the left the hazard function. In this case is represented by this notation, you can see here, and then the survival function here. So again, based on the values of the gamma, which is this value here, so you can see how different is the distribution here, and same for the survival function, which is again very interesting to understand the values provided by data. So there's something that is not really covered by the PPCR course, but this is another kind of different statistical analysis, which is called the Bayesian inference. However, some of you may know that the Bayesian inference is based on two probabilities, which are called the posterior probability and the prior probability. So the prior probability is based on the knowledge that we got of the distribution, or at least the theoretical knowledge, and then the posterior probability is based on the actual data that we have obtained. So the main difference of this approach compared to any other one is that it's taking into account the prior probability, which is the prior knowledge that we got of the problem. And this is something that we don't really take into account in more standard statistical analysis. So anyway, when we are talking about a prior probability, so we are talking, we have to talk about the beta distribution, and the beta distribution is this is the PDF for the beta distribution. So is so we have to plug some values for alpha and beta, and you can see here that it's a little bit complex as well because it involves the gamma and beta functions, which are represented here again. So you see how important are they. And then based on this, so you can see how different is the shape of the curve based on whatever you choose for the alpha value. So you see here the alpha value that I explained to you. The alpha value, the beta distribution basically has two components, the alpha value and the beta value. So they are plotted here in the equation, alpha and beta. And then so there is a number of combinations here for a number of beta values, for a number of alpha values here. For example, alpha equal to 0.5, alpha equal to 1, 2, 3, 4, and 5, and then for each graph I have represented the beta values for 1, 2, 3, 4, and 5 again. So you see that this number of combinations result in a very different distribution. So basically what the Bayesian inference does is choose whatever you think that that variable follows the distribution. So whatever you think in theory, okay, So, for example, you may think that prior to your study, you may think that your distribution of your data follow this funny curve here. So you just need to plug alpha equal to 4, and in this case beta equal to 2. So you see that you've got a number of options, and based on your knowledge you have to choose whatever the alpha or beta value is. And then as I said, the posterior probability is approximately equal to the likelihood times the prior probability, and the prior probability is given by these funny curves here, any of them, again depending on your alpha or beta value. Finally, there is something that is not covered by the PPCR course either, but this is actually quite important in epidemiology as well. This is called the Poisson distribution, and this is the probability mass function for the Poisson distribution. So basically the Poisson distribution is something that is used for counts of whatever you are interested. So for example, say when the count is or when the incidence of the event is very rare, for example in case of cancer, or in case of the rate of infection, on stuff like that. So you have to use Poisson distribution based on the counts of the number of people that are infected, or the number of people that have developed, for example, breast cancer. And again, the shape of the distribution, the shape of the curve actually varies a lot depending on the number of counts in this case. So this is what I'm trying to represent here. And again, this is done with this data. So this is what I wanted to discuss with you. As you can see, there are quite a few possible distributions that we use in statistics. I may have missed some of them, but I really think that many of the distribution covered here are important and very common in biostatistics. I hope you have found this helpful, interesting, and thank you very much for watching. So thank you and see you soon.